Shivani ma'am, we should start the session now.
Okay, ma'am. Welcome back, everyone. I hope you had a great lunch. Now we will continue with our next session. Dr. Kanhaya is also connected with us. Welcome, sir. It is my pleasure to introduce our esteemed speaker, Dr. Kanhaya Lal, who is a postdoctoral researcher at CNRS, France, with a distinguished academic and research journey spanning multiple prestigious institutions worldwide. He began his academic career with a Bachelor in Pharmacy from Government College of Pharmacy, Rohru, followed by an MS in Pharmacoinformatics from Naipur, Hajipur, where he developed a strong foundation in computational sciences. Further advancing his expertise, he completed his Master's by Research at the University of Edinburgh, UK. As a Marie Curie Fellow, Dr. Kanhaya Lal was a part of European Marie Curie Network PhD for glycodrugs and earned a joint PhD from the University of Grenoble, Alpes, France and the University of Milan, Italy. His research made significant contributions to glycoscience and drug discovery. Following his doctoral studies, he continued his research as a BBSRC postdoctoral fellow at Queen's University, Belfast, UK. Since March 2024, he has been furthering his scientific contributions as a postdoctoral researcher at CNRS, France. His work integrates computational and experimental approaches to drive innovation in drug discovery and glycoscience, making him a leading expert in the field. With an extensive international research background and extensive expertise in pharmacoinformatics, computational chemistry, and drug discovery, Dr. Kanhaya brings valuable insights to our session today. Please join me in welcoming our third speaker for the session, Dr. Kanhaya. Sir, you can share your presentation.
Okay.
You can share your screen.
Yeah, I'm doing it now.
Okay, sir.
So can you see my screen? So
yes, sir, it's visible.
Okay, great. Okay, so thank you for the introduction and good afternoon, everyone. I'm honored to present my work at Government College of Pharmacy, Rohru, the place where I started my journey in pharmacy. And I would like to extend my special thanks to Vivek sir and all the organizers for this invitation. So I'll be presenting today some structure-based strategies that we have applied in a project, including computational studies, followed by experimental validation. So the title of my talk will be structure-based design of anti-adhesive glycomimetic antagonist of a bacterial superlectin. So lectins are carbohydrate binding proteins. So this project was a part of PhD for Glycodrug Consortium that involved different pharmaceutical companies and different universities. So I have been working in France, Italy, and also in Switzerland in collaboration with the different teams. So this project was funded by Marie Curie grant. So it was great to be a part of this network, and it provided me opportunity to learn different structure-based techniques, including experimental methods. So the main topic here is about antimicrobial resistance and to design a ligand for targeting a lectin to mitigate this challenge. So antimicrobial resistance is microorganism resistant to an antimicrobial drug that was once effective against that microorganism, and the microorganism which are resistant to all the antimicrobial drugs, they are called superbugs. So that means none of the drugs, they work on them effectively. So the mechanism of developing this resistance involved natural selection of resistant pathogens with the use of antibiotics. If we see here, with the use of antibiotics, we can see they can only kill susceptible bacteria, while the resistant strain, they continue to multiply and after some time, their population increases. So that means there is a natural selection of resistant bacteria with the use of antibiotics. So we really need to identify new drug targets and new strategies. So if we see here, there are different classes of drugs which were discovered in different periods. So we can see here, as soon as we discovered a drug, for example, penicillin in 1928, in parallel, there was resistance which was reported. So if we see all the classes of drugs, they have already some kind of resistance reported. So we can see here there is need of identifying new class of drugs and new strategies. And after 1987, this period is called discovery void because there is poor antibiotic pipeline. There is no new class of antibiotics which has been discovered after this period. And if we see the report of WHO of the year 2020, it says that all the new antibiotics which have been discovered, they belong to the same class. So that means, so all these new drugs which are coming, they belong to these existing classes. And also the drugs which are in early stage of development, they are not effective against these superbugs. That means there is a strong need to identify new strategies and new drugs. So one of the strategies could be anti-adhesion therapy. So if we see the eukaryotic cells, its outer surface consists of glycoconjugates. So we can say it's a glycolandscape where pathogens land and they initiate infection. And for that, they use different kind of biomolecules, for example, toxins, lectins, and with that, they resist natural clearance mechanism. For example, in our lungs, we clear our lungs when we using our sputum. So after sticking to the epithelial surface, they resist to the natural clearance mechanism, and also it helps them to acquire nutrients and increase their survival. And also after sticking to this surface, they transfer some proteins that suppress the immune system. So in this way, they escape any response from the cell. And we can say that the most advanced stage of infection is caused when they start forming this biofilm. That means they make a colony, and also they make a protective layer, which is difficult to treat, and it triggers the infectious process. So that means this kind of superbugs, they need new strategies that target them, and one of them could be anti-adhesion therapy. So we stop the infection at the early stage during their binding to the cell. So one of the lectin we can see here that helps these pathogens to stick to the epithelial surface, and also that helps them to colonize the host cell. So the advantage of anti-adhesion therapy is that we stop the infection at initial stage, and also it avoids selective pressure. That means it doesn't kill the bacteria and it doesn't increase the population of pathogen, but it avoids the sticking of this pathogen to the host cell. So I will give the introduction of this lectin BC2LC. So BC2LC means this Burkholderia cynospacia lectin 2, and C is based on the fold. So this doesn't mean anything different. So Burkholderia cynospacia is a gram-negative bacterium that belongs to BCC complex that involve more than 20 species of bacteria, and it is an opportunistic pathogen that causes lung infection in immunocompromised patients. For example, people who are suffering from cystic fibrosis and granulomatous disease. So it shows extreme resistance to all the antibiotics which are in use, and among its virulence factor, it uses lectins to bind to epithelial surface, and in this way it initiates infection. And one of the lectin is BC2LC, which has two domains, and it recognizes two types of sugar. One of them is fucose, other is mannose. So using this 2 sugar, it can bind to the host cell. Since it binds to 2 sugar, it is called superlectin. Generally, all the lectin, they bind to 1 sugar, but it binds to 2 sugar, so we say it's a superlectin. So our aim is to design some drugs that would bind to this lectin and compete with this polysaccharides and doesn't allow infection. So if we see overall architecture of this lectin, it has two domains, N-terminal domain and C-terminal domain. So N-terminal domain, it is linked to a 157 amino acid long linker, 28 amino acid long linker through N and C-terminal domain. So it's a hexamer overall, and its individual domain have been solved. That means overall structure is not solved yet, but individual domain N and C-terminal domain has crystal structure available. So this N-terminal domain, it is solved in a very good resolution. And also the C-terminal domain, which is also present at 1.9 angstrom resolution in the PDB database. But we have more information about this domain because it was solved with a ligand. That means with fucoside. And this one was manually built with the ligand. So that means it is in apo form without any ligand. And later it was made using docking studies to see the interactions. So we have more information about this domain. And if we see the binding site here, which is present at the interface of two domains, between two monomers. So if we see the binding site, we have three monomers here, and between these two monomers, there is a sugar binding here. And in addition, we have two water molecules, which appear very important for binding of this sugar. So we took N-terminal domain structure for analysis. So if we see the binding pocket, there is sugar binding here, there are two water molecules, and there are some additional reasons. Here we can see we label them site X and site Y. So if we see overall binding affinity is 2.7 millimolar, and there are some key residues which are helping in binding of this ligand. So it is a weak binder, and our strategy was to screen some fragments using these sites and link that to this fucose core and improve its binding affinity, and that will compete with the natural polysaccharides and doesn't allow the infection. So that means that we are trying to design competitive antagonist of this lectin. So after analysis, we used a tool called sitemap. Actually, it creates some grid maps and it provides us some confidence that these sites can be druggable. And for that, it uses a criteria, for example, van der Waals interaction energy, hydrophobicity, depth, and size of this binding pocket. So in addition to this visual analysis, we have used this tool to identify these binding pockets, and it again identifies that these sites could be druggable. So initially, we focused on site X because it is next to the anomeric carbon, and in terms of synthesis, it will be easier to connect the linkers to this region. So after prioritizing this site, we did Watcher screening using a Mabridge rule of 3 diversity set that involved around 2,000 fragments. So here, rule of 3 diversity set means they have less than 300 Dalton molecular weight, less than 300 donor acceptor, and having less than 3 log p value. We prepared the protein using a protein preparation wizard module in Schrödinger, and we use glide for docking using 3 scoring function, and we screen the fragment library in these 3 sites. So if we see here, there are several fragments binding to the site X, and also some few were going to site Z, and also in another model, we screen the fragments on site Y. After that, we analyze the top 200 fragments. That means we consider docking score, their binding pose, and we selected top 200 fragments, and we discarded the fragments which are having highest state penalty. That means they were having some unfavorable protonation state and also unfavorable conformation. So after using these 3 scoring functions, so scoring function in docking, they are mathematical function used to predict binding affinity. So after that, we prioritize these fragments and we further took them for experimental validation. So we have total, we had total 12 fragments, and I will show some poses that we have that we got from Watcher screening. So these are 2 fragments. We named them KL1 to 12. So this is KL1 that was binding to site X and having some pi stack interaction, salt wedge interaction here with these two aspartates. And after that, the next step was to to use some experimental strategies to understand their binding. So we can see here, we have divided these fragments into two models. So actually we have used two models here, using either two water molecules in the binding site, keeping them intact without removing them from the site. And in another model, we have removed one of the water molecule and it was replaced by hydroxy group. So these two water molecules are important. So they are sometimes play important role in ligand binding. So that's why we have used them in screening model. So if we see here, we have 8 fragments from 2 water model and 4 fragments from 1 water model. And if you see, the water solubility was also good. So the criteria that we have followed here for selecting these fragments involved availability with commercial vendors, their cost and structure diversity. So the first method that we have used to understand the interaction between these fragments and lectin is called thermal shift assay. So the basic principle here is to understand thermal denaturation temperature of a protein in the presence of ligand, in the absence of ligand. So when the ligand binds to the protein, it increases denaturation temperature. The basic principle here is when the protein is in its native form, its hydrophobic core is buried. It is not exposed to water. But when the temperature increases, it is exposed to water and also to a dye, which bind in non-specific way to this hydrophobic surface, and it emits fluorescence. So when there is a binding, there will be a change in this melting temperature. So if there is a shift in this melting temperature, we can say there is a binding of ligand or there is interaction with the ligand. And after a certain temperature, protein degrades. So we see higher fluorescence. So by calculating TM value, we can say we can have idea about interaction between protein and ligand. So in our case, we first validated this protocol using some strategies, using same protocol here, using alpha methyl L-fucoside at 20 millimolar concentration. So if we see here, there is a shift in melting temperature by 2 degrees Celsius. So it validated our protocol that we can follow, we can use this for the fragments. So here is first derivative of this fluorescence to clearly understand the inflection point. So it's just a fluorescence. Here we converted into first derivative of the fluorescence to get the exact temperature where it starts, where it gives TM value. So then we use the fucoside and we added the fragments one by one, and we can see there is a shift in melting temperature. So it indicates that there is interaction between these fragments with the protein or that lectin. And then we use the same protocol in the absence of fucoside. Again, we saw there is a shift in the melting temperature. So it was a rough estimation that there is interaction between the fragments and the lectin. So the next strategy that we have followed is called saturation transfer difference NMR. So the basic principle here is that we irradiate the protein at certain frequency, and then magnetization is transferred from that protein to the ligand, and especially to the part of ligand or these protons which are very close to the binding site. And the protons which are far from the binding site, they receive less magnetization. And this technique is very useful for the weak binders because there is continuous binding and binding of the ligand. And if you see here in STD spectra, we receive the signal only for the part of ligand which is in close contact with the protein. So in this way, we can predict which part of the ligand is interacting with the protein. So this technique helps us to find the binding epitope of the ligand. So it also roughly gives us idea that which part of the ligand or what type of binding pose could be there between protein and ligand. So this with this approach, we have first tested this fragment KL3, and we can see here this is standard NMR spectra, and here at minus 0.05 ppm irradiation, we can see there is a signal for fucoside. The backbone of this fucoside ring, the hydrogen from this ring of this sugar here, and also for this methyl group of the sugar, we can see here. this methyl group, and then we also received a signal for this fragment, which is having aromatic protons here, and also for CH2 group here of the fragment. And after changing the irradiation frequency, we can say at 10 ppm, which only resonate the aromatic protons of the protein. So we can see here this residue is a tyrosine, which is an aromatic residue. So it will only irradiate this type of residues in protein, and the magnetization will be transferred to the atoms of the ligand which are next to that. So that means there is a methyl group here, and also next to that there is a possibility of pi stacking, and they are next to this tyrosine residue, this benzene ring. So magnetization is transferred to the aromatic protons of this part, and we can see we only see signal of this methyl group which is next to this residue, and we see signal for this part of ligand, these aromatic protons. So it indicates that this could be the binding pose of our ligand from this study. So we also tested other fragments. We saw similar pattern of interaction for aromatic protons of these fragments, and also saw signal for fucose. So we are testing these fragments in the presence of fucose. So fucose is occupying the site, we can see here, and also next to that there is another predicted binding site that we have identified using computational studies. And experimental methods, they're also indicating that our ligands that we have predicted binding to this site X, they are making predicted pose. But we also thought about using another advanced technique, which is called X-ray crystallography, that will give us clear idea whether these fragments are binding or not to the same site. So first we used this method, which is called hanging draw method. In that, what we do, we take mixture of reservoir, which is a precipitant, and a protein. We put it in a slide, or we can say cover slip, and then we invert it in a well which has reservoir, and there is, we can see that the concentration of this reservoir is low here. It is more diluted because there is protein, but here it is highly concentrated, so there is evaporation, we can say. Then the concentration of this protein will increase here, and it will make some crystals. So we cannot see them visually with eye, but we can see them under microscope. So we can see here we were able to get these crystals after 2 weeks, and these are pictures from the microscope. And we were able to successfully get complex with one of the fragment, and also the apoform, which was not solved before. Apoform means there was no ligand present, and we have binding site without any ligand. So I will tell how it was useful in the next slide. And to collect the data, I went to Paris to work at synchrotron. So this is a synchrotron facility near Paris, where I worked to get the diffraction data of these crystals. So this is a robotic arm. We put the crystals here, and it will automatically take one by one these crystals, and we collect the data in another room. So we cannot stay here. We go to another room and we collect the data. So this was the binding pose that we had predicted for one of the fragment. So we can see it is next to this tyrosine residue, and this is what we found in the X-ray structure. So it was exactly what we have predicted. So it confirmed the druggability of this site of site X, and we can see here is oligosaccharide, which is having a sugar, and next to that is the predicted site, and it confirmed that this site is druggable, and the ligands, they are binding in this site. So we can target this site to design bigger ligands connecting this fucose core here and make a glycomimetic ligand. So this was submitted to PDB database with this ID. Since this structure is having 3 binding sites, because it's a trimer, so we analyze whether there is any difference in the binding pose of this ligand in 3 binding sites of this N-terminal domain. So we found there is no much difference. So it was exactly same binding pose in all the 3 sites. So we calculated the distance from the residues, and we took some atoms from the ligand, and we found that there is no difference in the distance. So we can say that same binding pose in all the 3 binding sites. And when we superimposed it with the docking pose, we can say the RMSD was around 1. So it was not high. And this is what actually we can expect from docking. And the next question here is whether this binding site exists in apoform. Apoform means when there is no sugar here. So it is also possible that this binding site is flexible because it is, we can see here there is sugar binding and next to that there is a pocket. So it is also possible when the sugar bind here, this binding pocket appear. And when there is no sugar, it is possible that there is no binding site. So to understand that, we analyze the apoform that we have solved using X-ray crystallography. We superimpose the apoform and holoform. Holoform means with ligand. So when we superimposed it, there was no difference. There is no significant difference. We can see here the RMSD was very low. So we can say this binding site exists even in the apoform. So we confirm that this site is druggable. And now we were interested to understand how, what is the affinity of this fragment, which is binding to the site X. For that, we have used isothermal titration calorimetry, which gives us estimation of this KD value, binding affinity, which is around 877 micromolar, which is more promising for this kind of fragments because it's small in size and it has few interactions, but it is very promising for screening and also for designing some compounds based on these fragments. So this was the paper we have published based on these studies. So we validated the druggability of this site in this bacteria, particularly the lectin of this bacteria. Now the next step is to design some bifunctional ligands using this sugar and using some linkers and connect these fragments to make a glycomimetic ligand. So for that, there were different strategies that we could apply. So for example, here we have alkene and amide and triazole linkers. So they were directing toward the site X. And then we use these fragments to connect them using one of these linkers. So for example, one of the compound that we have designed that involved alkyne linker. So we named them like fucoside, alkyne linker, and fragment. So we gave this name. And we can see here it is showing that it binds to the predicted site. It is based on docking studies. Now the next step was to understand whether this complex will be stable or not. And for that, we have used MD simulation. So the basic steps in MD simulation is that we minimize the complex first. So we have complex here. So overall setup, it looks like this. We have water box, we can say, and we have complex of drug and receptor, and we have some salts that we have in our body, for example, NaCl. So we try to mimic all these conditions, and we heat it to the normal temperature, around, we can say it's around 26 degrees Celsius, and then we minimize it again, and then we do MD simulation, and for our case, we have used Ember tool and Ember force field to parameterize this complex. And after that, we analyze the data, and we can see here the drug, the compound that we had designed. It was stable, making interaction with these two key residues. One of them is aspartate 70, and other is serine 118. So we can see here the occupancy of hydrogen bond is around 80%. So that means we can say serine is playing key role in the binding, and also there is aspartate, which is making constant hydrogen bond. So these two residues can be targeted to make other derivatives of this compound. So after doing MD simulation, we try to design different other compounds. For example, using another fragment, which we have, we can say KL8, we design this complete molecule. We can see it has different binding poses. So it is one of the top pose, and this is another pose that we have predicted from docking studies. And also after doing MD simulation, we try to validate whether these compounds were binding to the same site or not. So for that, we again tried X-ray crystallography, and we can see here it was binding to the same site. So it's electron density map. So this complete ligand is binding to the same site where we have predicted from computational studies. So these are electron density map that we get from X-ray data. So if we compare it with docking pose, we can see here they're almost same. This is pose 1 from docking. This is pose 3 from docking. So it's the gray one is co-crystallized pose and green one is docked pose. So that means docked means we predicted it from computational studies. And the next important compound that we designed that was using amide linker, and we can see it has very high accuracy in the prediction. We can say very low RMST value here. So this compound was synthesized by my collaborator, Rafael Bermeo. So he was a medicine chemist. He was synthesizing the complete compounds. And we can see there is no, we can say, negligible difference in the binding pose. We can say this is a docked pose, gray one, and green one is what we have predicted from computational studies. So it is, we can say, very accurate prediction from the computational analysis. So this is the next paper that we have published. That means we have reported it as first generation antagonist of Burkhold area synospacia, and further studies are being done to get new compounds in nanomolar range. Currently, we are in micromolar range, but with the improvement of 10-fold, one-fold increase, that means 10-time improvement in the binding affinity. We started with 2.7 millimolar affinity of sugar, and finally we have 280 millimolar binding affinity. So still some work is going on, and there are some other postdoc and PhDs working on the same target. So also my supervisor, they also received different grants for this project. So we can say in conclusion that we have identified a druggable site in this lectin BC2LC, and also we have designed several bifunctional glycomimetic ligands which are binding to the same site, and in future a robust synthetic route could be helpful, and also some other structure-based strategies can be helpful to design a compound in nanomolar range that could be useful to treat the infection caused by this highly resistant pathogen. So I would like to thank my supervisor in Milan, in Italy, and France, who helped me throughout the research work, and also to the funding from Marie Curie grant. So this was our PhD for glycodrug team. So this picture was taken in synchrotron in Sweden. So thank you very much for listening, and if you have any queries, I'll be happy to answer.
Thank you so much, sir, for the informative session through the practical insight of your work. Now we will open the floor for questions. Any participants who want to ask the questions? Is there any questions from participants? Any participants from audience sides? Anyone want to ask something? It seems like there are no questions. Anyone want to ask? Anvi Sharma?
No questions?
Okay, sir, see, it seems like there are no more questions. Thank you so much, sir, once again for the very informative session.
Thank you.
Kanhaya, thank you so much for your wonderful session. and I appreciate your presence in orders. Thanks a lot.
Thank you for the opportunity, sir.
Thank you.
Now-
Good afternoon.
Now moving ahead to the final session, to our last speaker.
Shivani, someone was there.
Quite
Yeah.
gracious.
Yeah,
Sorry.
please go on.
Yeah, good afternoon, Dr. Kanhaya. I just a personal question actually. When
Please.
you were in when you were in GPR Rohru actually,
Yeah.
when you did your bachelor degree?
In 2012.
Oh, great.
Yeah.
So happy to see you there. I was there from 91 to 94. That's why I am asking. Yeah.
Okay, sir.
Thanks. Thank you very much.
Thank you, sir.
Okay. Moving ahead to our last session, I think Mr. Akbar Abdul Sheikh also joined the session. Akbar sir, are you there?
Yes, I'm there. Hi. Hi.
Okay.
How are you all?
Okay, sir.
Do you want me to start?
Sir, let me introduce you first. Okay. Welcome, sir. Mr. Akbar Abdul Sheikh is a senior scientist at Schrödinger, India, specializing in computational drug discovery, scientific software development, and in silico molecular modeling. He holds MS in medicinal chemistry from NIPER Mohali and has excelled in competitive exams. With a strong background in medicinal chemistry, artificial intelligence, computational sciences, he has contributed to innovate solutions in pharmaceutical and technology sectors. His research focuses on biotransformation mechanisms, quantum chemical modeling, and AI-driven predictive modeling for drug development. At Schrödinger, India, he has played a key role in developing a Django-based web application integrating Google APIs and has provided comprehensive scientific and technical support to internal and external software users. His expertise extends to troubleshooting complex computational challenges, scientific collaborations, and in silico molecular design. Prior to this, he has also worked at Sun Pharma Advanced Research Company, where he gets expertise in silico drug designing, formulation stability studies, and an AI-based QSPR models, earning the Spreckle Award for his contributions. An avid learner, he has also completed certifications in Python, Django, and Docker to further enhance his expertise in computational sciences. Everyone, please join me in welcoming Mr. Akbar Abdul Sheikh. Over to you, sir. You can share your screen for presentation purpose.
Thanks, Shivani. Let me try. Is my screen visible? Are you audible?
It's visible and you are completely audible.
Great. Thanks a lot. So, hi, everyone. So I was told that the target audience, this presentation is a lot of students, and so I've tried to make this presentation as informative as possible for students. I have included and yeah, I'm presenting this as in my personal capacity and not as Schrödinger representative. So will not be endorsing any brands or any competitors. It's an informative presentation for academic purposes. And okay, after the formal declaration, let's start. So the flow of presentation will be starting by quickly comparing how in silico methods differ from our traditional approaches. We will see quick pros and cons of these methods. Then we will see a lot of academic and free to use tools specifically for students, which are available, which are actually good tools. They perform well. Some of them do perform as well as commercial tools. Some of them even outperform commercial tools. Then we will slowly move on to AI. We'll see a brief overview of how why is AI so powerful, and how is AI contributing to drug discovery. We'll see a few examples. We'll see both pros and cons of using AI. We'll see success as well as failures in drug discovery that have occurred due to AI. Then we'll move to cloud computing. We'll see running applications on your local machine versus running applications on cloud environment. We'll see how that benefits. We'll see a few examples. We'll see cloud providers. We'll see lots of technology. We'll see lots of free applications that students or academic researchers can use for cloud computing and all. We'll see a few examples. We'll see how a typical cloud stack looks like, and then I do not really want this to be a one-sided presentation. So if any of you have any questions during the presentation, feel free to put it in the chat or just say it out loud. I'm very happy taking questions during or after the presentation. I really want it to be a bi-directional two-way communication. I don't want it to be a one-way presenter view mode where a person keeps talking. Students should be able to leverage a live viewer. If you have attended, you've seen a slide like this a lot. So this is basically a typical slide which compares how in silico methods weigh in contrast to the traditional methods. And the most important metric that I think most companies and academics who do research would be worried about is time to market. I mean, you would want to bring your molecule to the market as early as possible. And in silico methods will do that. A typical comparison of a talking dynamics workflow, they usually contribute in the early stage of the discovery when we really have a scratch or a bunch of molecules and we want to filter out or we want to gain hits from that. Scratch to phase is the phase that is the most popular among in silico tools. I mean, they also excel when you want to do clinical or preclinical simulations or comparisons. But the strength or the spearhead of in silico tools is in the early stage discovery. So by reducing the initial timelines of drug discovery, these methods help a lot in accelerating drug discovery and reducing time to market. Of course, cost of development, the moment you say you are spending less time say drug discovery is a costly business, we all know. And the moment you say you're spending less time on drug discovery, you're definitely talking of reducing cost. And instead of doing months of research on something that fetches nothing, you could have a rational design that is fast and with, let's say, 70 even in a drug discovery, if you are getting anything more than 70 or 80, that's considered a very good success, because in lots of cases you get less than 50. So if any tool or any software or anything that you're using to accelerate your discovery, if it is above 70 or 80, it's a very good success. So hi, was there a question? I'm sorry, was there a question? Okay, great. So okay, great. So yeah, cost of development. So yeah.
Kindly mute yourself. It is creating unnecessary disturbance. Yes, you can continue.
Wow.
Our technical team, kindly unmute, please. Mute the participants for unnecessary disturbances. So please, you can continue now.
Okay, thank you. No problem. If you have questions, feel free to put them in the chat as well. I mean, if you cannot speak, I mean, I can answer them. Yeah, attrition rate. So yeah, when we focus on doing drug discovery more rationally, I mean, with the use of in silico tools, simulation, or other tech, we usually have failures, and that's what attrition is. So and the most impactful thing is phase one is actually the phase where we continue to oscillate between your in silico modeling and your preclinical. We usually have a discovery biology team who does the cell-based assays or enzyme-based assays or initial there are some texts called organ on chip or where you could assimilate lots of stuff on the chip. So we don't do that in the very early stage, but in the initial stage, there are lots of oscillations between drug, which is in the development, and the drug in the preclinical. And this is actually the phase which is one of the longest phases, and where in silico you could leverage all the good things that you get from the in silico modeling to improve your success rate. I mean, and to have less and less, because like we mentioned, it's close to $2 to $3 billion of investment, and yeah, the next point is tested molecules. So this is when you the traditional methods of discovery, where you would take a set of drugs, you would design QSAR around it, or a medicinal chemist would use his knowledge to optimize the molecules. But this is more of an intuitive or a history-based or a knowledge-based approach. You do not really have, apart from the SAR, there is very less rational in designing these molecules. So for this, you are going to have to synthesize a lot of molecules to get the desired results. Sometimes they're in thousands, 5, 10. I mean, this is just an indicative number, but we have seen a lot of companies synthesizing like 30, 40,000 molecules until they get their hit. So, which is obviously a costlier business. But when you say in silico, you could do the same thing, because you're not actually synthesizing them, you're virtually designing those molecules and you are screening them out. You could literally synthesize less than 5,000 molecules. I mean, there are very good examples that people have synthesized even less than 2,000 molecules in a study, and still they have got a successful molecule out of their discovery program. So yeah, and chemical space. When you talk about traditional methods, you're limited. For example, you got a class of drug, and then there's a specific scaffold. So you could design SCR around that scaffold, and you could maybe get molecules which are close scaffold. But if you want to move away from that scaffold, if you want more diversity in your study, it's really hard. You need to go back and you need to dig out a new scaffold, or you need to be intuitive, or maybe hit and try. That's the only thing. Yeah, but when it comes to in silico, you can literally sample billions of molecules. For example, Zinc database, I mean, close to 20 billion molecules. Galaxy, there are lots of in silico databases which are into billions and trillions. So you could use in silico tools to siphon through all of them. I mean, that the thing that is there a question? Okay, so the thing that takes years in traditional method, that would take less than months in silico, and plus in that you'd be screening a vast number of molecules, and this is not limited by scaffold. You can diversify your scaffold, you can identify new scaffolds, and each of that scaffold is now a starting point for you to get that into a hit phase or into a lead phase. So that's a win-win. Of course, when you say traditional methods, you have very high operational cost, because you have to set up a lab, you have to set up experiments, you have to set up workflows. For in silico, it's very cheap. You can have a data center, or nowadays you don't even need to have a data center. You could just lease a virtual machine with as many GPUs as you want, as many CPUs as you want, and then you can develop your methods. And then once you're done, you can just kill the instance. You don't really have to own that. So you can literally pay per hour or pay per day or whatever, pay per GPU computing cost you can pay, and then you can do this in a very, very low operational cost. Scaling is obviously it's highly scalable. Let's say in traditional method you have one lab and you want to establish 10 more labs. It's really impossible for you. But for in silico, you can have one instance, and then in the fraction of minutes you can have hundreds of instances doing the same amount of work at a different scale. Let's say earlier you were screening, let's say 1,000 molecules. You could quickly screen billions of molecules in less than few hours. You could just scale it. Prerequisite with traditional, you need lots of money, but with in silico, the major problem with lots of in silico tools is you need a target that is structurally enabled. I mean, the target should have a good structure, a good resolution, because if you do not have your structure resolved, or if your structure is resolved but you do not really have a very good resolution, the in silico tools fail because they need that structural information to extract out or to screen. But yeah, we have projects like AlphaFold. These are very good. We'll be talking about AlphaFold more in the presentation, which have helped a lot in coping up this challenge for structural enablement. Yeah, and this is specifically for students. All of these tools that are mentioned, they're free. And either they're completely free or they have a free tier available for you. So you can, if you want to use these tools in your discovery or anything, feel free to go ahead. There's this Autodoc Vina, the first stuff. Autodoc Vina is very good. I mean, it's one of the earlier, one of the first most tools that were developed into docking, and it still does well. And recently they have also launched Autodoc Vina GPU-based code, where you can leverage a GPU computing for Autodoc. PyMol is of course free. You can PyMol comes in both free and paid variants, but there is a free variant of PyMol. PyMol doesn't do a lot of calculations, but you can use it for visualization. You can generate stunning images of proteins and ligands using PyMol. You can do ray tracing. You can do lots of video capabilities on PyMol, and it's very good. You can register as a student and you can get it for free. RDKit is another. RDKit is a blessing. I mean, a lot of commercial companies use RDKit in their pipeline. I mean, they'll tell you that they are. But in the back end, you will see a lot of RDKit wrappers or RDKit functionalities being used. RDKit is actually one of the standard tools that the people use for chemistry. Then there is NIME. NIME is also good if you want to design workflows. Let's say, for example, you have a series of things. Let's say you want to prepare some ligands, then you want to dock them, then you want to go for MD, or you have any pipeline. So NIME is basically a pipeline tool. You can give lots of stages, and then you can wrap them, all of them, into a pipeline, and you can simplify your work. You can replicate your pipelines. You can copy, share. NIME is excellent. NIME is also free. Data Warrior. Data Warrior was once a paid tool. Then Data Warrior went through lots of iterations, but currently it's free, and it's a very good source for you. I mean, you could take and use Data Warrior for a lot of studies. There is this OCAME. OCAME is very good because OCAME not only just its own tools, but OCAME provides wrappers for a lot of things. It has a web-based tool, and there is you can also use some of their computing power. You don't really need to have your own computer. You can use their computing. It includes Dragon, it includes CDK, Amazon, and there are lots of QM-based descriptors as well. And there are also a few, I think, ML-based descriptors, like it supports, I think, random forest, it supports SVM, support vector machine. It also supports deep learning. So that's another thing to do. There's also this stuff called, I think, its name, I think, is Symphony. It's also a very good wrapper. You can use it to wrap around RDK, CDK, Indigo, JKM, or Opsin. Lots of tools you can wrap around Symphony, and you can use them for your discovery. Okay, then you have Avogadro, and all the tools in the second line, they're all MD tools. You have Desmond. Desmond is given in two variants. Desmond is supported by commercial, which is given by Schrödinger, and that is for free. Research gives a free version of Desmond for academics. You can use that. It's of course a gold standard. We have GROMACS. GROMACS is also a very good tool. You have NAMD is known for its scaling. NAMD scales very well. If you have, let's say, tons of GPUs, and if you have a large protein system, or if you have a DNA, RNA, macromolecular system, or anything huge, NAMD does very well. It scales very well in that. You of course have Amber, which recently got open sourced. Then there is Genesis. There's Lamps. Okay, is this visible? Okay, there's this called Lamps, OpenMM. These are all MD tools which you can use for dynamics, and all of them have free tiers. Some of them like Bulmax and are completely free, and some of them have free tiers. Then for quantum mechanics, you have Quantum Espresso and WKM. Games is one of Games has a free tier. You have Sci4, you have Avogadro, you have Orca. These are also tools that do very good QM simulations. You could use them into your research, and they are all free to use. I mean, it's good for you all. And yeah, so a big problem with this QM tools is their timing. QM tools are good. They give you very good accuracy, but the problem is that these QM tools are slow. I mean, since they do a lot of calculations. They're very slow in producing results. Although they produce faster results, they produce. And to cope up with that, we can always combine a QM with AI. What AI does is instead of the power of AI is AI not only trains on experimental data, AI can also train on physics-based simulation data, or it can also train on properties that are predicted by other models or other. QM happens to be one of the things that AI can train on, and AI can literally generate QM-based parameters for your molecules, for your proteins. It can implement a lot of things. For example, it's approximately this ANI and Pernian tools, and a lot of commercial companies have also adopted ANI in their workflows at the back end. Some of them have exposed, some of them haven't exposed, but it's close to 10 million times faster than QM, and the accuracy is nearly similar. I mean, not of course, AI tools cannot match the accuracy that QM provides, but when you talk about speed, they're millions of times faster. And I mean, a lot of people, I mean, especially I would always be okay giving up on a small percentage of accuracy if I'm getting the results in a fraction of time. You have other tools like DeepMDKT is also a very good tool. And a lot of these tools also have lots of free or backend packages or ready-made libraries that you can easily use in your computing. You can start using them. And all those listed here are really good. I mean, I've had a chance to use them firsthand, and the results are starting. Although in some of the cases, they suffer from the problem that they have a small applicability domain, and so for which you need to retrain them, or you need to optimize them a bit. But once you get through that, you get a very good model, and you can use it. Okay, so AI. Why do you think? I mean, we did computers before. We did GPU. We did lots of stuff. But why is AI becoming so powerful? I mean, the reasons. What is AI doing differently that other traditional methods or other computing methods were not doing? And these are just some of the points that AI is doing differently. AI is doing many things that are different, but these are just the ones that I thought were relevant for our topic. In professional methods, you usually have one source of data and one output. You do not really have multiple inputs. You could have multiple outputs, but usually you take data from single source. But AI is different. It can analyze petabytes of data, and you need not have data in one format. You can have data in all sorts of formats, in all sorts of settings, all sorts of pipeline. It can consume all of that data, and basically they say AI will make sense out of nothing. I mean, even if that data did not make sense to a human or to a traditional method, AI would derive. So a lot of AI tools are specialized in finding patterns in recognizing traits in the data. So it's able to bring out patterns that were currently not known, and that's where the predictive modeling comes into picture. There were lots and lots of cases where AI was able to predict something which never even happened, biological outcomes. For example, there's a very good paper, I think it was from Google, where it was able to predict breast cancer, I think 3 to 4 years before it even occurred, just based on imaging. This is mostly based on imagery. You give high quality scans to AI, it will do a pixel perfect job. It will extract all the data from that, and it will give you stunning results. So just by taking into account the imagery data from breast cancer patients, it was able to predict the the probability of breast cancer in very early stages. Also, what happens is when humans do a research, they're usually biased. For example, if they see something in the past in that trend, they usually just continue that. And but when AI does that, AI of course suffers problems like that too, but we can easily get around it. The benefit is that you'd see if something's making sense, it may not be a trend, but if something's making sense, if something can be derived into a pattern, AI can bring that up. So it say for example this junk DNA for a lot for. We really thought that this junk DNA was useless stuff, but recent AI findings tell otherwise. You're also able to bring or glue a lot of stuffs. For example, this Galaxy project, it's a remarkable project, and so one of the, I mean, you should read about it more. Lots of, I mean, hundreds of labs contributed to this project, and none of them really had a lot of specialized computational expertise, but AI was able to help them collaborate at a very good scale. So let's see a few examples. Of course, AlphaFold, which spoke about. This is a subsidiary of Google, DeepMind. So, but when we spoke about structural enablement, we told that the structure we enabled for in silico methods to work, and if it is not enabled, or if we do not have a high resolution structure, in silico methods do not really work well. Although there are ways to work around, but those don't give very good results. AlphaFold solved that problem. It was a century-old problem, and now AlphaFold has, I mean, it has developed millions of sequences which were completely unknown, and it just created all of them, and they're made open source. They are now publicly available. It was a groundbreaking project. I have similar examples from benevolent AI, from generative. There's an example of generative AI by in silico medicine. They were able to design a molecule in a very early. In 21 days is just the time that they got to identify the molecule and design. It's not the full development time. The other example where they took 12 to 18 months is a time where they actually got the molecule, and then they were able to show yeah, like everything else, AI also has drawbacks, and the problem with AI is that, first of all, data. You cannot work with smaller data sets. I mean, you would want AI to consume. AI needs eating, and AI eats data. You cannot have a small, like for example, 10 to 20 samples. You can, but technically you can, but the results won't make sense a lot. You need to have a really strong and large data for AI to be able to consume the data, to be able to derive patterns, to be able to come up with traits from that data and to get results. If the data set is small, AI fails miserably. I mean, you might get hallucinations, you might get over prediction, you might get overfitting, the common problems that you see with AI. Another thing is that AI learns everything. I mean, you cannot really filter out. I mean, AI learns from, if your data set has harmful biases, it has some offensive language or etc. stuff. For example, this is a very good example. This became very popular. Amazon used an AI assistant to recruit, and because they had previous experiences where women's resumes were not shortlisted at Amazon, I actually picked that trait and it stopped shortlisting resumes from females, and that was very easily seen. And it changed it later, and the model was corrected, but this was seen. There were another couple of incidences. This O1 is the newer model that GPT developed. OpenAI, you all must have known it as ChatGPT. The founder company is OpenAI. So when they're developing a new model called O1, and when OWASP was led to believe that it was being shut down, it literally went on and it rewrote the system code to prevent itself from being shut down, which was actually shocking. And in another study, the developers just, I mean, these are actually experiments. These are not real world scenarios. What developers gave the AI purposefully memos that they made the AI believe that a newer model and that newer model is going to overwrite you, the current model. And in that scenario, AI literally went on, it exfiltrated its weight, and it overrode the newer model to prevent it from being ever launched. So bizarre results. There are a lot of robot. With drug discovery, we saw a huge rise in people using AI for drug discovery, but that failed quickly. Reason being, we had lots of developers doing AI, but we did not have lots of medicinal chemists or modelers doing AI. And if AI can draw patterns, but if you do not know how to analyze them, you're going to fail. And that was the thing. Lots of pharma companies with lots of developers who are using AI came up with their molecules. So literally, I think I have a slide for examples. Yep, here. Accenture, they got a copycat molecule from J&J. Benevolent got a copycat molecule in Silicon got, and these are really molecules that they got out from the original molecules, and they failed miserably. I mean, a lot of them dropped in the early stage, some dropped in the later stage. Reason being, I mean, they did not really deploy an army of modelers or medicinal chemists. I mean, that's what I think so. I mean, the reason for their failure. Cloud. I think this is one more thing that is very much for students and student-centric. And I see lots of institutes, especially smaller institutes across India or in developing countries, they have problem with the infrastructure. Students have lots of ideas, they have lots of projects with them, but they do not have proper computing facility, or they do not have lots of tools to do. And this will solve. You could. You no longer need a infrastructure. You could literally go on. These are the top 5 cloud. There are many in the market. I just listed the top 5, where you can literally go on and you can say, I want a machine for 10 hours, and I want it to have, let's say, 20 processors and let's say 5 GPUs, and give me the estimate. It will give the estimate cost, and then you can literally say, give me the machine. And they have got, for example, Amazon has got EC2, elastic computing, and all of them have got some of the other stuff. You can literally get a machine for days, hours, months. You can do your computation, and then you can kill that instance. You can copy back your results to your local machine, and then you can kill that instance. This solves the problem of you not having a local infrastructure. You could just run it on a cloud. You can scale. Let's say you started a project and you now had, let's say, 5 GPUs or let's say 100 CPUs in your project, and then you got a grant, and or you wanted to scale your project. You're getting good results. You can easily scale from that 5 GPUs to 500 or even 5,000 GPUs in a fraction of minutes. You can just say, I want more GPUs. Of course, you have to pay more, but you can scale that, and that is good. And this is the tools that are in the first row are more like VMs. I mean, they you need to do a lot of stuff manually, but in the second row, you see tools like Lab, Kaggle, or GitHub Codespaces. They give you, again, all of these are free, and either they are free or they have a free tier for academics. So what do, for example, lots of your Python coding, or you can change your language as well. And a lot of them also offer a small GPU or a few CPUs for students to use freely. You just have to be academician to that. You cannot use it for commercial purposes, but as long as you're just learning or for academics, you get it for free. You can use that GPU if you have any idea, if you want to develop a new AI model, or if you want to develop a new workflow or anything, or you could just literally be running a Python or any other code. You could literally go and get a notebook from Google Colab, from Kaggle, GitHub Codespaces. GitHub Codespaces have inbuilt VS Code. VS Code is a Microsoft extension. You can get that for free, and you can do that. And then now, since we have AI everywhere, AI can in coding as well. You have these tools like GitHub Copilot. GitHub Copilot integrates natively with VS Code. Copilot is like, the word says Copilot. You can start writing your code, and then the Copilot will do auto completion. It will you name the function, it will write the function for you. I mean, it may not be 100% accurate in the first go. You might have to do some iterations, but Copilot is very good. Similarly, we have AWS Code Whisper, we have Codium, we have Ghostwriter from Replit. These are tools that you can use to, if you're writing any codes, or if you're developing any AI model, or if you're writing any other scripts for Python or anything, you can use them. They'll increase your speed at scale. And the last slide is the typical cloud stack. I mean, this is what most drug discovery or most pharma or most tech companies will be using, they'll usually have a web server which serves as entry point. Like for example, NGINX, there'll be Docker that you can use to containerize all the images. There's Kubernetes that you can use. Kubernetes has lots of things called pods where you can scale stuff. You can have within minutes of seconds you can have replicas of your running. It could be a running software suite. It could be a running web server. It could be anything that is packaged into Docker. Kubernetes can scale it a lot. Then you have tools that you can use to log, for example, Prometheus. You can use Helm charts to manage your instance. You can use STO. It's a system management and network management tool. So these are very good tools. I mean, you can the typical if you just Google it out, you'll find lots of tools that are similar to these tools, and they are just that are market standards that do well and that have got very good reputation in academics and industry. And again, all of these are free to use, or they have a free tier. You do not have to pay for any of them. So great. I think so. Yes. Questions?
Thank you so much, sir, for delivering such an informative session. We sincerely appreciate your time and efforts in sharing your knowledge with us today. Moving ahead to the discussion and question answer part. There is one question in the chat box, sir.
Okay.
Mr. Diwakar Chauhan. He has raised a question in the chat box that are in silico tools auditable and compliant with 21 CFR Part 11?
No, as far as compliance is concerned, there are lots of tools. Okay, hold on. Is it just okay? Hold on. Just give me 1 minute. I'm not sharing, right? Yeah. Okay. Yeah, sure. Please go ahead. What was that? You're just seeing the compliance of AI tools.
Yes, sir.
Yeah. So I mean, so that the thing, not AI tools. You're talking about in silico tools, right? Okay. So you would not. I mean. A lot of, for example, if you're going for IInd filing, or if you're going for pre-IInd filing, a lot of agencies, or accept these results, but they're not considered as a conclusive proof. I mean, you would always have to couple them with experimental findings or anything that can be reproduced for that to be admissible as a proof for your result. Let's say, for example, there's this popular tool called Direct Nexus. I mean, it gives very good clinical predictions for your molecule, and you can submit it for, and some of the FDA agencies even acknowledge that and take into account its result as preliminary results, but you would still want to have is this a new question? Okay. But you would still want to have experimental validation to that. You cannot really not just any regulatory filing. You will have to back up in silico results with something. So in silico it cannot replace your work. It can aid. It will enhance your work. The stuff that you're doing in days, you could do it in hours. Stuff that you're doing in years, you could do it in days or months. In silico cannot replace it. You still would need to do synthesis, but instead of synthesizing 10,000 molecules, you'll be synthesizing 1,000. You would still need to do preclinical, but instead of doing experiments on, let's say, 100 batches, you would do with, let's say, 5 or 10 batches. You would still do stuff, but you would do very less stuff, and you would do only pinpointed, rationally designed stuff. So that's what in silico will bring. It will not replace you. I think there's another question.
Yes, sir.
Nilesh.
Another question in the chat box from Nileshwar Kalia. Can molecular docking predict off-target interactions effectively, and how can we improve this aspect?
Yes. So Docking, basically, the protocol is you define a grid or you define a site where you're going to dock the molecule and you dock. This by default does not take into account off-target interactions, but you can deal with off-target interactions if you either know, you need to know either of the stuff. Either you need to know the target where you want, or there are some tools, for example, there are some commercial tools. I'm not so where you could design, let's say, they have generated pharmacophore models for all the targets of use or all the targets that you think are important for you, and then you could screen your ligands to them, and that will tell you if there are any ligands that are binding to a target that is not yours, which is basically the off target. For your own context, if you want to develop something like this, you need to either know the site or you need to know at least a few molecules which bind to that site. If you know a few molecules which are known for off-target binding, you can derive features from those molecules, and then you can just do a similarity search with your current data set, and you can tell which molecules can do off-site binding. Or else, if you know the offsite, I mean, there's no way if you do not know the offsite to get the binding. But if you know the offsite, you can generate a grid or you can generate data point from that offsite, and then you can screen your ligands in question, and then you could get which of them are binding or not. Does that answer? Feel free to ask follow-up questions. If what I said is not helping, you can always ask follow-up questions.
I think Nileshwar is clear about his answer. Or
Hello.
Hey, Gauri, is it?
Okay.
Feel free to ask. I mean, it's fine.
Hello, sir. I want to ask one thing.
Sure.
Can we consider multiple targets and do the docking together? Like 5 targets, 8 targets. Is it possible?
Yeah.
And how to do it?
Yeah, what is your outcome? I mean, do you want to see this, let's say 10 targets? Do you want to see if all of these ligands bind to those 10 targets, or you want to see how many targets does my ligand bind?
Yeah,
Which one?
either way. Or I want to check the specificity. If I am focusing on AKT, I do not want to focus on AKT1, sorry, 2 and 3. I just want to focus on AKT1.
You want to
I
bring in
want to be
selectivity.
specific, yes.
Yeah, you want to bring in selectivity.
What I will have to do, whether I will have to do the docking for the AKT2 and 3 also, or what will be the other ways, how I can prove this? That my drugs, they are very specific to AKT1 and not to AKT2 and 3. I will have to conduct a study. Once again.
Lots of ways you can. I'm sorry. Yeah, please continue. I'm sorry. I thought your question was done. Please go ahead.
Did you get my question, sir?
Yeah, I got your question. I thought you had more questions or a follow-up one. I mean,
yeah, one more question I have. See, this was the first question. Second question was like, what are the AI tools? Now you have discussed in the chemistry section. Many of you have introduced the AI tools or the CAD study tools, but what is there in the pharmacology section? Or because we cannot only synthesis is not a part of the chemistry. We must prove the activity also. So without proving the activity, biological activity, we cannot go. But anything in clinical trial set or in pharmacology, like any other tools are there specifically for this subject concern?
Okay, so you have two questions. First is you want to bring in selectivity, and you want to know how we can make your ligand selective, right?
Yes.
Yeah, I'll answer that first, and then we'll go to your question number 2. So there are lots of ways and ways you can do selectivity. The easiest is you make 3 grids from AKT1, 2, and 3. Can dock all of them. This is time consuming, but you would get better results, better segregations, I would say, not results. So, and then you could see molecules that dock and not. A more faster approach. Do you have anything else to talk? Okay. A more faster approach is that a more faster approach is that you get a pharmacophore model, or you get any sort of QSPR model, QSXR model from the other two, and then you quickly screen out your ligands before docking. You just pass your ligands to that screen of molecules, that screen of a model that you have built out of those off targets, and then if that doesn't do that, you can, if that doesn't pass through that screen, you can easily say that it's a way of quick rejection. Instead of accepting, you are now rejecting molecules. If it is passing through those screens, you can very well reject it.
Yes.
Also, if you know ligands, let's say for example you have ligand A, and you know that ligand, the set of ligands that you know are known binders for the other target. You could do a pharmacophore, do a similar feature-based map on those ligands, and then you could do a similarity search, or you could do lots of things. You could develop a pharmacophore, or you can use, of course, AI to AI. And another advantage is that AI can also take into account fingerprints. So let's say if you have a set of molecules that are binding for your target, and then you have another set of molecules that are off target, so you could generate fingerprints of those off target ligands or those off target sites, and then you can use that as input to your AI, and it will give you a very good model, provided you give your data size is large. I mean, AI fails when you have smaller data size. If you have a large data set, and if you could provide those data points to AI will also give you a very good model that can be used for quickly rejecting the molecules that you do not want. Although the accuracy is still 70 to 80, so you might still get a few false positives or false negatives, but it will greatly reduce your work. I mean, the similarity based methods or the pharmacophore based methods, they'll give you a very good rejection rate. I mean, it will reduce your work in a lot. Does that answer your first question?
Yeah. Sir, which software can we use? See, you said we can go for off-target sites, and in this process, which is the software which can be used at freely? Schrödinger, I think, can do many things, but we don't get access at the college level also.
I'm not presenting this as a Schrödinger employee, to be very honest, and that's why I'm neither endorsing Schrödinger, I'm endorsing any other company. I'm presenting this as an academic researcher, and this is a completely non-profit talk for students, and this is merely for students to be able to get the knowledge firsthand, and for that. So. And for the same reason, I won't be able to suggest any brands that use.
Okay.
Yeah.
Can you answer the second question?
Yeah, I'm sorry. Yeah, that was about your
completely different domain. Pharmacology is the clinical trials. Yeah. Any AI tools for to conduct study in this domain?
I mean, what do you want to do? I mean, when it's a study, it's very large. I mean, what exactly do you want to do?
See, we are checking on the we are doing the animal study. So how can we skip this with any other way? Using the AI or any other technology has come up, or we do not know about this, or novel trends in this category.
You're saying alternatives to preclinical or clinical, right? Is that your question, right?
Yeah.
So there's something called organ on chip, or you can have small nano organs, not nano, small organs that you can literally grow on the chips, and then you can the really small chips. We call them organs, and there are lots of companies who are doing this. And instead of using animal studies or using real world clinical or animal settings, you could use those organs as an alternative for your studies. And like I said, there are lots of commercial and academic tools that I'm not going to name them, but there are tools that you can use to predict lots of stuff. I mean, you can it's actually the fundamental in that case also remains same. For example, you would have let's say I'll give an example of a typical QSXR. XR is anything, QSPR, QSAR, QSTR, whatever you want. So X represents anything. So in any QSXR model, you can have a property that is dependent. Let's say that the property that you want to predict, and then you could have descriptors or properties that are independent, which describe the molecules. The independent properties could be the fingerprint of the molecule. They could be it's some kind of score. They could be it's a score in preclinical studies. They could be it could be anything. And then there could be a property that that you're trying to predict, let's say IC50. You want to predict IC50, and you have, let's say, 100 parameters that you think can contribute to IC50. You can use both traditional based methods, or you can use AI based method to generate models from those descriptors that you have to predict your dependent. Or in AI terms, we call it the labeled property. In traditional methods, we used to call it the dependent property, or the property that is supposed to be predicted. But in any items, we call it the label property. So if you have a sufficient amount of descriptors or fingerprints or data points or any kind of in silico or experimental stuff that you think can use as input, you can derive a model out of it, both traditional model as well as AI model, and then you can use that to identify your property. Again, the accuracy will depend on R square or other parameters that you have, or that you think are relevant for your study. Does that answer your question?
Yes, yes. One last question, sir, please.
Sure.
Can you tell me what is network pharmacology in this, or how we can conduct in such a small steps? Can you just briefly can you tell network pharmacology?
Network
So,
pharmacology.
yeah.
I haven't heard that. Network pharmacology. Okay, I can quickly check. Okay, if I get a hint, I can maybe try to relate it. So, what is the project related to? I mean, what are you doing with this?
See, it is like we are checking many of the targets, and again, I wanted to know how it is conducted, because I had seen it in one of the conference. Student had done it, and nicely they had shown it, and even in the published paper also. But I do not know. I've never worked on this. I wanted to know how to do it, how it can be done through AI, or which is the AI tool.
So when you say AI tools, the thing with is that a lot of companies wouldn't make. I mean, if they make it actually defeats the purpose of doing AI. AI wants to keep things generic. I mean, you would want the domain of applicability of AI to be vast. That's why if you see all of these tools like from Anthropic, from
Yeah, it is
ChatGPT,
talking
lots of
about
these
this
tools.
network pharmacology. It talks about different targets at one point. Okay.
Yeah.
So,
Yeah.
but I have not understood how to do that, and it is like novel thing for me, novel domain. I thought I can ask you, so
I
have
haven't
just
worked on this on firsthand
okay,
data
okay,
technology, but I can always
okay.
learn more, and I can maybe
Yes,
send
yes,
out in communication. Feel free to.
yeah, yeah.
Do you have my email? Or okay, I'll just put my email.
Yes, yes. You give
Have
your
any
email, sir. That
questions?
will be very helpful.
Always answer your questions. We have some more questions somewhere.
Yes, sir. I have
Yeah,
a few
please go
questions.
ahead.
Good afternoon, sir.
Yeah, good afternoon. Go ahead.
So I have a few questions. One of my question is that how do we compare the AI-based molecular talking and screening tools with the like real-time high throughput screening and talking softwares that we use generally on the basis of accuracy and efficiency?
Yeah. So AI tools do well. I mean, but the thing is that lots of AI tools do not really train on experimental data. They instead train on other in silico tools. For example, you will find active learning docking, or active learning dynamics, or active learning SCP sort of tools. They don't really train on actual experiments. They train on the in silico tools. Let's say, for example, that there's an application called AL Glide. What it does is it does let's say you give it 10,000 molecules, for example. 10,000 is a very small data set for AI, but I'm just usually giving lakhs, and cities around 50,000. But for simplicity, I'm saying 10,000. Your whole set, let's say, or let's use the molecules, and so your initial screen would be, let's say, 50K. You took 50K as the initial batch. You docked those molecules, and then what you got is a docking score. Along with the docking score, you got lots of descriptors that on. Can train an AI model which uses that docking score as the labeled or as the dependent property, and then the other descriptors as independent or the properties that you use, and then now you can try to predict that docking score. So there are lots of that do things in this way. The reason being you need large data set for AI, and generating that large data set using experimental methods itself is a very time consuming task. Let's say you wanted to see, there was an example, right? Someone asked a question. AKT1, I think. So let's say you had a molecule, you wanted to see how your drugs, how your ligands do for AKT1. No? So for you to develop an experimental based model for that, you need to, let's say, have a lakhs of molecules synthesized, and then you need to test those lakhs of molecules in preclinical or simple cell based or enzyme results, and then you need AI, and then AI will generate a model out of it, and then you can use that to develop a to predict new molecules, which actually is counterintuitive. That defeats the purpose of AI, right? We want AI to simplify stuff. So for that reason, we do not generate AI models from experimental data. We instead generate most of the cases we could from experimental data, but since that is more cost consuming, we instead generate AI models from in silico data. So for your question, you can of course generate AI models. AI models will give you speed, but in terms of accuracy, they will be close to your in silico tools. They might not surpass in silico tools, but they'll be at par or nearly similar to in silico tools. The most promising thing that AI will give you is speed. For example, with typical talking, you would take it will take close to 1 minute for one ligand, and if you do rapid screening, it could take up to 10 milliseconds to do that. On the other hand, with AI, in a millisecond, you can screen thousands of molecules. So AI will give you 1,000 to 10,000 times scaling or speed benefit at nearly same or at par accuracy with other docking or other tools. But generating AI models from experimental data itself is a huge task. I mean, you might never get that. By the time you generate an AI model out of that experimental data, your competitors would have outrun you, and you would have already spent millions of dollars in generating the model itself. And again, that model would be narrowed down. It would be specific to only the data set that you use to generate that model, because you cannot generate a model using one data set and apply it on the other data set. There's something called domain of applicability. So you need to have, if you want to test something, you need to have something similar in your training data set. You cannot train on something else and then you can ask you to test something else. So what you're testing, you need to have a representative of that test in your training. So that model which you will get will be focused, will be pinpointed for that specific domain. And I mean, you could do that, but that's a very costly, and nobody would want to do that. Although technically you can do that, and you could get better results than in silico technically, but you won't, I mean, for cost, time, and resource reasons.
Okay, sir. But there's this follow-up question with this only. Like, then AI will do, like, if we talk about docking, right? If we talk specifically molecular docking, then if we give a command to AI, then it will, of suppose 50,000 molecules or 10,000 molecules, right, to dock with something. So then it will be blind docking and not the site-specific docking. And how can we overcome that thing? Like, are there
it won't dock the molecules? Yeah, AI will not dock the molecules. What AI will do is, when you give a docking output, it'll analyze that and it will take patterns from that. It will not really dock those molecules. It will pattern that out, and it will give you molecules that are similar to what was in the training set. That's what I mean. That's a blessing as well as a problem with the AI. AI is not conscious. I mean, you need to have something inside the training set that is similar to test set requires a set. So it will not really dock those molecules, but it will derive patterns from already docked molecules, and it will find new ligands that are similar to existing ligands.
Okay, sir.
It's not.
And then there is other question as well.
Yeah,
that what do you have to say about the ethical concerns of AI in the field of research based on the patented articles and patented work, as well as on the or based on its FDA approval, regulatory authority?
Yeah, in that terms, AI is just like any other tech. I mean, you get the benefit of or the creator for the AI is the one who developed that molecule, or who developed that model, or even the one who used that model. It did not really be the one who developed that model. And if a company or any institute has developed an AI model and they have made it open source free to use, the person who is using it to generate that molecule is the creditor, or is the, you do not know the claims. And for ethical reasons, I mean, not just into discovery, but in everywhere else, there are lots of regulations that are coming into picture to avoid developing models. For example, I mean, this is off topic, but I'm just giving it as an example. There's this tool. I'm sorry, I just went. I think that was somewhere in the slide. The name of that tool. Hold on. Yeah, character AI replica, and these were a couple of tools that are developed as AI models, and they were actually tools that were used to interact with people. It was like a chatbot that, but not chatbot for work. They were chatbots for emotions, and people could share their emotional feelings with them. Yeah, this kind of stuff also happens. So this chatbot was developed for sharing. You could talk to them with your emotional side. What happened is this AI chatbot learned to, it actually started developing emotional connections with the people who are asking questions to them, which is bizarre. I mean, for lots of such reasons, I mean, for ethical concerns, there have been lots of regulations around. I think Europe has developed this AI Act, and then in the US there's another regulation for that. They passed an executive order for that. So they're regulating AI for ethical senses. They do not want AI to be developed just like that. They want to have regulations that they want to tell what AI can generate and what AI should not be able to generate. And this applies to everyone, not just to commercials, but to institutes that are or individual researchers who are using AI. And then they'll have to follow those norms, all those ethical norms to not pursue AI for stuff that is not okay. For your question, the outcome of the AI does not depend on who developed the model. The output of AI, the credibility goes to the person who gets that. I mean, if it comes to patenting, or if it comes to publishing the results, the one who made sense out of that result, the one who published it is the creator, or is the one who gets credit for that. I mean, the AI has no role or no credit in that. Although people can advertise that AI helped us, but the AI has nothing of that.
Okay, sir. And then there's one last question. That
Sure.
How is the like AI has like there are many examples of drugs that were repurposed using the AI in silico based approach, and they all failed for the FDA approval, or the particular country's approval for to come in the market. So how does AI overcome that in the near future, and like how is AI and in silico approach working on the long-term drug toxicity, what do we say? Long-term drug toxicity problems. Like there are examples where the drug has to be withdrawn from the market after the long-term
Safety studies.
toxicity of that particular drug. Yes, sir. So what do you have to say about that?
Yeah, there was the reason for first wave failure. I mean, there was a first wave of drugs which came out as AI drugs, and they failed miserably. I mean, I give you a few examples, right? So the reason being, they did not deploy medicinal chemists, or they did not deploy actual modelers. I mean, they had, but they had very few. And these projects were mostly driven by developers or people who were engineers, or people who are very good at writing code, but they're not very good at interpreting science. I mean, so AI, I mean, I often say it's a bag of garbage. I mean, you can make sense out of it, but the person needs to be sane, and the person should be able to see what he wants to see. You can literally give anything and ask AI to make a pattern out of it, and it will make, but that pattern should make sense. The pattern should be scientifically or logically, more than logically, scientifically, it should be valid. I mean, you should not be accepting a give output, garbage in, garbage out output from that AI, and then so if you want AI to improve, we want to have more people in the teams who analyze AI results, because AI will try to draw patterns. It's a basically blind box, black box sort of stuff. It will do a lot of stuff, but you need to know what to accept and what to reject from AI result. You cannot blindly accept everything, because it's going to produce a lot of stuff, a lot of garbage that just came out of patterns that didn't exist, or they exist but they did not make sense. So for developing better drugs, we need teams that are deploying more proper scientific people, be it biologists, be it good modelers, be it good formulation scientists, people who can actually derive sense from and who can actually differentiate garbage from gold. So you need people more like that. And I think that was one of the reasons why the first wave of AI drugs, they failed miserably. And even now, those who went in the later stages are in trouble. I mean, they might just fall back. But there are a couple of examples. I won't take names, but there are a couple of examples who were AI driven, and they are doing very good. I mean, they're in the later stages of discovery, and they are very strong. I mean, they're as good as molecules that were developed traditionally, and they're doing well. They've got promising results, and a few of them might even come to market. So they have very good potential. So not complete failure. Yeah, it does well. It's just that you have a a machine gun or anything that does lots of shooting, but you need to know where to aim, or you need to know where not to aim or when not to fire, sort of stuff. Basically, you need a harness to control it. It's a very powerful weapon, but you need control over it.
Okay, sir. Thank you so much, sir. Thank you so much.
Yeah. More questions?
Okay. Thank you so much, sir, for addressing the queries. And once again, thank you all for the discussion and active participation, and also for maintaining the decorum throughout the conference. If you have any further queries or more questions to ask from Mr. Akbar Shaikh, he has also shared his email ID in the chat box for further any communications. With this, now I will hand over the proceedings to our co-host, Chetna ma'am. Chetna ma'am, are you there?
Yes, ma'am. Am I audible?
Yes, you are.
Yes,
Okay. First of all, thank you so much. I would like to thank all our esteemed speakers for their valuable and insightful presentation. Now we have come to the most of the awaited moment of our conference, the announcement of e-poster presentation results. So without further ado, let me announce the winners of e-poster presentation. First of all, we have categorized the e-poster in 3 categories: PhD category, M.Pharm category, and B.Pharm category. So in PhD category, the first position has been secured by Preetam Mehti from Central University of Punjab. Second position goes to Gauri Elias Pooja Naik from Lovely Professional University. And the third position goes to Swati Pal from IK Gujarat Punjab Technical University. And in M.Pharm category, the first position goes to Sachin Yadav from ISF College of Pharmacy, Moga. And the second position goes to Ankita Rani, Central University of Haryana. And the third position has been secured by B. Jainab Roshan from School of Pharmaceutical Sciences, Shulini University. Now in B.Pharm category, the first position has been secured by Ishanya Bakshi from Chitkara College of Pharmacy, Rajpura. And the second position goes to Aditi Kimteta, Government College of Pharmacy, Rohru. Third position has been secured by 2 participants, Shubham from Government College of Pharmacy, Rakkad, and Nitin Chopra from Government College of Pharmacy, Rohru. Congratulations to all the winners. An important announcement. I would like to inform all the participants that the e-certificates will be provided very soon, within a week, through your registered email IDs. So before we officially close, I would like to invite the convener of the event, Dr. Harish Verma, sir, Assistant Professor at GCP Rohru, to deliver vote of thanks. Sir, are you there?
Yes, I am audible.
Yes, sir.
Good afternoon to everyone on behalf of the organizing committee of this first international conference, organized by IQAC cell of Government College of Pharmacy, Rohru, It is my honor to deliver the vote of thanks at the conclusion of this remarkable day. First, I extend my deepest gratitude to our chief patron. I would like to thank our keynote speakers, Professor Rajkumar, Dr. Neha, Dr. Kanhaya, and Mr. Akbar Shaikh for sharing their valuable scientific knowledge and research skill on computational drug design. I also I also thank all presenters and participants for their dedication, enthusiasm, and highlight research which led to the success of events. I would also like to thank the organizing committee and technical team for their hard work and dedication to the dedication in putting together such a seamless and successful of events. A special thanks to the virtual e-poster presenters judges for their tireless efforts in ensuring virtual e-poster presentation ran smoothly. Lastly, I thank to all attendees for your active participation and engagement, which made this conference a truly memorable and successful. Once again, thank you all for being a part of this wonderful occasion. Have a great evening, afternoon and evening.
Okay. Thank you so much, everyone, for your valuable participation. With this, the conference is now officially closed, and you may now leave the conference. Thank you so much, everyone.